This right here. So hi, let me pull up my slides here so we can all be on the same page. So thank you for joining us today. We are delighted to welcome you to our fall event for all of you that are able to attend in person and online. We are Georgia Tech Astrobiology. We're the collective network of members of the Georgia Tech community that are interested in the search for life in the universe. It's an interdisciplinary network encompassing people representing a whole bunch of different fields, from aerospace to planetary to biology. And we have researchers, we have enthusiasts, and we have you that are in the room and online with us today. You are all members of our community, and we're so excited to have you here. We want to thank CSTAR, which is the Center for Space Technology and Research, for helping us co-sponsor this event today. And I also wanted to point out that after the talk that we're going to be hearing today, you have the opportunity to come continue to network with the astrobiology community. We are having our fall social, which will be outside for all of you that are here in person today. It's outside where we will have food and trivia and prizes out on the patio. We have new t-shirts, which a lot of us are wearing in the room today already. So even if you have one of the yellow ones from previous years, we have new ones that you're welcome to come pick up for free from us. We have stickers. It'll be a great time. So join us from 4 to 6 today for that. So with that being said, I would love to introduce our speaker today, Dr. Sarah Stewart Johnson. We're really happy to be hosting her today virtually to hear more about her perspective on astrobiology. She is a Provost Distinguished Associate Professor at Georgetown University, and she has been a member of the NASA Spirit, Opportunity, and Curiosity rover teams. And she is additionally the author of a nonfiction book from 2020, The Sirens of Mars, about the exploration of the red planet and the search for life on Mars. So I would love to pass it over to Dr. Johnson to tell us more about astrobiology, and we can hear from her. Let me stop sharing on my end of things, and you are more than welcome to take over, Dr. Johnson.
Oh, thank you, Tatiana. Can everybody hear me okay? This is all work. I was trying to decide if I should leave my phone on. I went ahead and turned it off, but I have this colleague who gave some lecture just into the ether for an hour and hadn't realized it disconnected. So hopefully we'll have no internet problems. But thank you so much for that really, really kind introduction, and just for having me. This really means a great deal to me. And you guys have just an incredible program at Georgia Tech. So many great people and colleagues and friends, and I'm just really excited to be here. So I guess I will share. I'll go ahead and share my slides. All right, can you see? Let's see, does that?
Yes,
yeah, look okay. Okay, perfect. Well, I am again just so happy to be here, and what I thought I would talk about today, this title, it's Contending with the Truly Alien, Agnostic Approaches to Life Detection. And I think I'm going to talk for about an hour, and then hopefully we'll have lots of time for questions. And so please don't hesitate to ask questions, especially students. It'd be great to hear your questions. And I put in, so I'm at Georgetown, and I put my email address there. So if anybody has any questions that we don't get to, or if there's anything else you want to chat about, please don't hesitate to just drop me a line. Okay, so just to sort of get right into it. So I had this great conversation with the graduate students before we began, and just sort of thinking about how I found my way here, and this question, this big question, are we alone in the long night? This is one of these questions that keeps me up at night, that I just find so captivating on so many different levels. And I've sort of made it the center of what I do as a scientist. This kind of cornerstone of my work is really around the idea of looking for biosignatures or traces of life. And so really, how best can we go about looking for these kind of fingerprints of life beyond our planet? So that's sort of what my scientific research is all about. And I know most of you guys are really captivated by that question, just like me. So what I do at Georgetown, really do a lot of work kind of thinking about how to search for these biosignatures in planetary analog environments. I've got sort of a molecular biology wing of the lab and an organic geochemistry wing of the lab. And so we do this work going out to these analog environments, lots of Mars analogs, but also ocean worlds analogs. And we think, like, I mean, we're actually sort of looking for traces of life, but we're also thinking a lot about sort of training and technique development. And so, you know, trying to understand things like the limits of life. That's sort of a big question, of course, for many of us in astrobiology, like how far can you push life and it still survive? Because some of these, you know, planetary places that we're exploring, some of these environments, you know, are quite hostile, at least in terms of life as we know it. And we'll get more into that in just a second. But, you know, here's one of we actually have 3 field sites, you know, sort of looking at lava tubes, which are these kind of basaltic lava tubes at craters of the moon in Idaho, near Mauna Loa in Hawaii, and also in lava beds in California. And, you know, so there are these incredible sort of channels that have been carved by this magma, and they leave behind these places where you can look for life. And there are a lot of these lava tubes up on Mars, right? So Mars has lower gravity, and mineralogically, you know, some of these flows are very, very similar to those kinds of basaltic compositions that we see up on Mars. And so this is an exciting place. I think one of the near surface repositories potentially for even extant cells or even biosignatures at some point, they could have been habitable. But you think about environments like these and the Atacama Desert and other places where you're really pushing life, you know, like in these environments, total darkness, we've got sort of chemolithoautotrophs, you know, that are finding a way to even just like sort of on the surface of bare rock, you know, find a home. So we do some studies like this. We think about things like the persistence of life, you know, so we have biomolecules that go back billions of years, like into our planet's history. And so, you know, thinking about how recalcitrant are those molecules. I was talking to some of the graduate students earlier about some studies that we've got going on in Antarctica and the dry valleys, and also the Untersea oasis, which is in Queen Mud Land on the other side of the continent. But, you know, Antarctica is a really fascinating place, you know, no birds, no plants, no mammals, just entirely really dominated by this microbial ecosystem. And, you know, you had much more climate conditions in lots of Antarctica, and those, it's gotten colder, it's gotten drier, much like Mars, and thinking about how can biosignatures and even cells themselves kind of survive over long periods of time. These really cold and arid conditions in some ways are just fantastic for preservation. And so some interesting studies there. These microbial mats in Lake Untersea are really phenomenal. They grow up to 70 centimeters tall, some of the largest stromatolites, or these are kind of modern unlithified stromatolites, but really interesting analogs for the early Earth and what, you know, life might be like in some other places. And then what we do is we try to kind of apply what we've learned to space exploration. So working, you know, with missions and like kind of thinking about sort of the next generation of missions and instrument development and those types of things. That's another kind of thing that we do here, at least as part of my lab group. So, you know, one of the things that we have is we've really developed as an astrobiology community these really robust approaches for life detection. So we know how to go into, you know, a new environment and identify these well-established, widely accepted features that are associated with terrestrial life and the signatures of biological processes. So some examples of those, you know, particular classes of molecules and isotopic signatures, patterns within the molecular weights of fatty acids, and other lipids, biogenic structures, biogenic textures, biogenic minerals. You know, there are all of these very, very, very robust ways that we can sort of look for different things that are associated with life, these kind of canonical biosignatures. And you know, we can do that in extreme environments. We can go out to places and use a lot of these kinds of techniques, look for these sorts of things and find, you know, evidence that life is there or was there in a different form. And this is just another example, another field site that we work in are these acid salt lakes in Western Australia. These are, this is kind of an overview of what some of them look like. These pictures here were taken by my colleague, Kathy Benison. On the left, these are from Mars, and these are some similar sort of features that you see in these same acid salt lakes in Western Australia, where you get, cross-bedding and different cool sedimentary structures, and these bugs, displacive crystals. You can see they've been dissolved away here, but you see the same sort of gypsum crystals here. And this place, like this is like pH of battery acid. It's got these distinctive minerals that we see on Mars, like jarosite and alunite, and we can use what we learn about life to kind of make predictions about which suites of minerals, say, might be the best targets for life detection. But so we can do that kind of in extreme environments. We can use the kind of sort of canonical biosignatures and such to go back to the early rock record of our planet. That's how we've reconstructed much of life on Earth over the epochs. And then, sort of pushing this out to other worlds. And so here's Curiosity. One of the instruments on Curiosity is the sample analysis of Mars, or SAM instrument. It's the size of a microwave. It's in the belly of the rover. It's looking for organics, the sort of building blocks of life as we know it. And it's been fun. We found all kinds of simple organics there, as well as kind of all of the sort of essential elements for life as we know it, the schnapps elements. This is sort of showing some chlorobenzene and dichlorobenzene, also thiophenes, like, and more exciting stuff to come, so that's been really, really fun. Okay, so like we were really good at sort of using these more traditional canonical biosignatures. And there's some really extraordinary benefits, of course, for looking at for what we call life as we know it. So for one thing, we're starting from what we know. So it's a lot easier to kind of interpret the data because we really understand what we're looking for. We have the sort of model, the exploration model in our mind, and we understand what these biosignatures look like. Signals can be really, really rich in information. And we also have a stronger understanding of the effects of taphonomy, which in some environments is really critical. So the same way, like here on Earth, you can sort of have, say, we don't find a dinosaur when we go look in the Rocky Mountains. We might find a dinosaur bone, and from like a dinosaur skeleton, we can sort of reconstruct what the animal was like. We do that on a molecular level, just with molecular fossils. So, we might not see the original biochemical. It will go through this process of molecular fossilization. You'll lose some functional groups, maybe some double bonds will break. We will still, like, even if you kind of lose the fingers and toes of the molecule, you can still see the backbone and from that sort of reconstruct what it is. And also, especially as we've kind of gotten our feet wet and started our search for life, when it comes to places like Mars, it's just as similar to Earth as it gets. This is a place with, it's a similar size, it has similar inventory of volatiles, and the sort of starting materials here in the inner solar system were pretty similar attributes. The silicate crust, it was also once protected by a magnetic field. Of course, we know that by looking at the ancient highlands in the southern hemisphere, and we see these remnant magnetic field stripes. That's of course very important. We have all this deleterious radiation sort of coming in, but of course it was deflected early in its history. And we know that Mars was warm and wet early in its history. At least periodically. We know that from looking at the geology, all kinds of geologic features that were carved by liquid water, all kinds of hydrated minerals, and all sorts of things that are very indicative that Mars had a much different past than it does, than the planet, than the conditions today. Mars is also so close, it's just right there. And so, there is this really exciting, intriguing possibility of shared ancestry that, life was carried either from Earth to Mars or Mars to Earth. Of course, the latter path being more likely with the gravitational well of the sun. And so, the work of folks like Ben Weiss at MIT and others, they've taken meteorites from Mars, they've carved into them, they've shown that they haven't necessarily been heated above these sterilization temperatures. Some of them arrived on really quick trajectories. And we know microbial life can take a beating. It can just sort of wedge into a crevasse. It can hold on against all odds. And it can like live in a rock, like all these endoliths all over Antarctica. There are just all kinds of possibilities. So like, it's also one of these things that people say, like, say if you lose your keys at night, the first place, of course, you'll want to look is underneath the street lamp because it's so easy to see there. And so all of this, I think, makes a great deal of sense, starting with what we know, thinking about life as we know it. But I do think it's really important to be careful and cognizant as we go about this whole endeavor of the limits of analogy. So both in terms of where we are on the spectrum of understanding life, just like as we know it, and also sort of more broadly, just thinking about our ability to even imagine things beyond our current thinking. And so one example that I think is nice and illustrative is we think about like kind of where we are just kind of in our own understanding of biology back in like the late 1960s, early 1970s, when the Viking missions were being designed and planned, the first landers on the surface of Mars. And, we just had this really different kind of understanding of biology and microbiology back then. So for instance, like we did not know what we know now, that just the vast majority of life on Earth, when we think about all the different biodiversity, the vast majority of life on Earth that can't just be cultured and in a laboratory. And we didn't realize that before the advent of genomics. We just didn't realize that there's all this life out there that we just kind of couldn't see because we couldn't grow it in the laboratory. And so in the life detection missions, like there's this one kind of famous experiment on Viking. It was Vance Oyama's chicken soup experiment. And it was this whole idea that you just take this really nutrient-rich broth and you throw in some Martian soil and it'll be like a little petri dish and you'll get growth. Like we just didn't understand that wasn't really even a great model for understanding how life works on Earth, and that like tons of the life here on Earth wouldn't sort of grow in those conditions in the laboratory. So I just would argue that, if we were designing those experiments today, we would probably design them differently, just based on our own understanding that like so much of life, just kind of won't grow in sort of a nutrient-rich broth. So we're always kind of evolving our understanding, learning more and more and more and more. But I think this is also, the limits of analogy when we think about like just all kinds of possibilities for life that are just really hard to imagine, and it's, we were talking about, I was talking about the graduate students, it's so difficult because we see these worlds and, it just seems like time and again, we've just been bowled over by their indescribable foreignness. We see these worlds and see worlds like Mars and it just feels like, oh, I could, fall down, I could scrape my elbow, I could pick up this rock, I could, you feel like, oh, this is like just like Arizona, like this is just some place that's eminently familiar and recognizable, but of course they're just indescribably foreign and so alien. And so we really have to sort of be aware of that. And we've seen that kind of time and again with sort of our just understanding of how these planets work and how they're put together, like how their tectonics work with their atmospheres. And I just think it's so important for biology as well. Okay, so how is it then? Can we go about looking for life, not as we know it, but life as we don't know it? And so this is a really exciting time, so this big report, an astrobiology science strategy for the search for life in the universe, came out a couple of years ago. And one of the big recommendations of all these experts was, the search for life beyond Earth. It really requires more sophisticated frameworks for considering the potential for non-Terran life, and NASA really should be supporting research on novel and or agnostic biosignatures. So I would also say that it's a really exciting time to be considering this question. For so long, especially with our Mars exploration strategy, it was just follow the water. Let's follow the water. And we found so much water, so many golden swimming pools worth of water and all kinds of evidence for water in all kinds of different forms. And then, we're finally embracing, like with the Curiosity rover, and this idea of exploring habitability, but with those results, all the building blocks of life, all those essential elements. We landed in Gale Crater and we found not just acidic water, but like circumneutral water, the kind of water, as John Gottsinger got up and said, if you were standing on this planet billions of years ago, you could have a drink of this water. We've found, an environment that is certainly habitable. And now with Perseverance and, also with Rosalind Franklin, which we hope will go at some point, moving now to this new phase, we're actually seeking signs of life, not just the context, not just the habitability, not just the water, but really looking for signs of life. So we're finally doing life detection for the first time, since Viking, and it's really super exciting, a very exciting time to be a Mars scientist interested in astrobiology. And of course, it's not just sort of NASA anymore and ESA and the Roscosmos, we're getting this widening range of involvement. All these new players, sort of jumping in a bigger way than they had before, thinking about Mars exploration. So we're sending new missions from, all kinds of different other parts of our globe, private companies like SpaceX and others sort of thinking about getting into the exploration game as well for planetary exploration. So that's really exciting. More data. And we're also getting improving technologies, these huge benchtop instruments that are so difficult, like advances in miniaturization, like we're seeing these things shrunk down, new ways of even taking big data sets and condensing them to like data streams that we can actually handle from the far reaches of the solar system. We're just seeing lots of new ways and new kinds of instruments that we are able to send now that, a decade ago would have just been impossible. And then like kind of new targets, I know this is still like very debated. Like, is this, some sort of a radar reflection from some minerals, or is this actually, down in the South Polar layer deposits? Like maybe there's this kind of subsurface briny lake, which would be so exciting, to someday drill into that if it exists. There's still lots of debate, of course. But we're getting like new places with, all of our incredible reconnaissance that we could potentially target as part of the search for life. Where like, this is mostly sort of thinking about Mars, where life might be ancestrally related. It might also not be, if it's there at all. And then this is also just another really big, exciting thing. And I think especially important when we're thinking about life as we don't know it. And that's expanding deeper into the solar system. So we've got these like amazing new rockets, like the SLS, we all, Monday was the first day of school for my little kiddos, and we got up at 8:33 and we were hoping to watch the launch, and hopefully this will go soon. But this is this enormous rocket, bigger than the Saturn V, and it's just going to be the most powerful launch vehicle that's ever been built. And when you couple things like that, like with advanced power systems like solar electric propulsion technologies, it's enabling us to reach these places on timescales that are like kind of reasonable to do planetary missions. And so of course this brings in this whole realm of ocean worlds, these icy moons of the outer solar system that we can explore and we can think about. And they're far from what we sort of used to consider, or what we still in some cases consider the sort of habitable zone, but like could totally be habitable. So agnostic tools and techniques, I think they can really enable us to search within a broader framework for life, especially the further we get from Earth. I think the more important these are, and especially as the environments get really different. Mars and Earth are still pretty similar, but other places. So some of those targets, we'll just do a walk through some of my favorites, but this is the first glimpse of Europa from July of 1979. This is from the passing Voyager 2 spacecraft. And so the spacecraft traveled 500 million kilometers and like comes back with these images, and it's just like, what are those? Are those troughs? Are those ridges? Like, or maybe it has something in common with Earth's plate tectonics. I think it was just at this moment of discovery, it was just so confusing. You've got this incredible amount of technology. We've sent Voyager 2, it's bringing back these images, but it was just so confusing. And sometimes you get this amazing advances and you think it's going to be crystal clear, and then you just can't figure it out. But this is around the same time. So in 1979, the first paper appeared in GRL saying, questioning whether there might actually be liquid water on Europa. But I mean, how could there be? It was so far out. It was so cold. How could there possibly be liquid water? But of course there is. There's this huge under ice ocean, and it's all of that tidal heating because Europa is constantly contorted by Jupiter, which is 300 times more massive than the Earth. So you've got this kind of crazy thing where it's locked in this tidal resonance with Io2, and you get all this heat generated, all this kind of friction. And all of that contortion, creates this huge ocean, and we think that ocean, maybe has been there since the creation of the entire Jovian system, so it could be very, very, very long lived. So 2 years later, you get Galileo going out, and even though it's having trouble with its antenna, it was able to confirm that really Europa was best explained by having this liquid water ocean, and that icy terrain had, these kind of plates that were sort of moving around. And the magnetometer also detected this induced magnetic field between Jupiter and Europa, and the easiest way to interpret that finding is really with this salty global subsurface ocean. So now we have this beautiful high-res images of this planet's surface with these incredible bloodshot eyeball appearance with all these radiation processed salts there on the surface. But of course, with Europa Clipper and this sort of next generation of instruments, it's really exciting to kind of be thinking about the possibilities. And yeah, so here's Europa. It's projected amount of water, more than the water of all of our oceans, caught there underneath the ice on this little moon. It's kind of constantly morphing and changing in terms of its surface. And again, we might have even 2 or 3 times as much water on Europa as we do here. So, we don't really know exactly how thick that ice shell, but it's, it's probably more than 15 kilometers. So it is kind of hard to access, but, we know very amazing things that even though Europa is bathed in radiation, there are microbes that can even survive, like these Deinococcus radiodurans that can survive inside the cores of nuclear reactors. And, we also know that there are what we think these plumes, so here are some of these Hubble observations, comparing transits from I think 2014 and 2012, showing these possible geyser activity near Europa's South Pole. And so, we do it seems like there's also been more recent evidence building for plumes on Europa, which might give us a way to sample without, melting through or even landing. If we can go, get some of this plume material, which we think is the underwater ocean coming up. But of course, no doubt about geyser activity up on Enceladus, another great target. So Enceladus is tiny, a seventh the size of our own moon, about the size of little England, but it's got this fresh, clean ice, one of the brightest objects in the solar system. We get the E-ring, this diffuse. It's very, very wide, and this is being created by all of this material that's being blown out of these cryovolcanoes, these geysers, through these tiger stripes on the South Pole. And so this is a really exciting other target, we've got 500 pounds a second of this briny stuff being thrown out through potentially 100 different geysers. So lots and lots of exciting possibilities. We might have hydrothermal vents here that we would have there where, the sediment water interface underneath that icy shell comes together. Of course, one of the big theories for the origin of life on Earth, maybe it was, sort of thermal environments on land, maybe it was deep in the ocean and hydrothermal vents. Very fun news from the Planetary Decadal Survey coming up with an Enceladus Orbolander being the second big priority. for planetary exploration in the next decade. So the idea that we could orbit and then potentially land and look at some of this ice, the material that's been sort of created by the snow of the brine, would be so exciting. And then finally, Titan, Titan with its pale hills and its dark rivers and its hydrocarbon rain. I mean, this is just such an intriguing world, minus 180 degrees Celsius, the largest moon of Saturn. It's the only place other than Earth where we've got clear evidence of stable bodies of surface liquid, but of course it's hydrocarbons, it's ethane and methane and not liquid water. So here's this place, it's got clouds, it's got weather, it's the only moon we know of with a thick atmosphere. We've seen the surface from the 2005 Huygens probe. And we've got Dragonfly coming up, hopefully with a launch in 2027, arriving in 2034. And hopefully, like, this is a radar image from Cassini. Like, it really begs the question, if you've got a non-water world like Titan, this kind of gasoline world where there's hydrocarbon lakes, what might the sort of molecular and polymeric building blocks of life look like? What might those be? And so I think that these places are also really exciting to kind of think about this question. Okay, so now I'm going to tell you about some of the science we've been doing as part of this project called the Laboratory for Agnostic Biosignatures, which is a NASA ICAR project, which is an interdisciplinary consortium of astrobiology research. This is sort of the successor to the former NASA Astrobiology Institute. These ICAR projects that are situated within these research coordination networks. And so in full, the network for life detection is the one that's sort of relevant for this work. There are 5 different RCNs. And so this project has been going on now for a couple of years, and it's been one of the really kind of exciting things that I've loved thinking about, because I do think like this is one of the deepest sort of intellectual challenges that we have. I mean, it's almost like trying to imagine some color that you haven't seen before, and it's getting at the heart of this thing, like, how is it that we can really contend with a truly alien? And how do we go about searching for indicators of life without presupposing an underlying biochemistry or any kind of a particular molecular framework? And I just think it's like a big, exciting, interesting challenge. And it's kind of, I just think it's so interesting to think about. And I've been having so much fun doing it. So quickly, here's our team. So like the former NAI and NASA Astrobiology teams, this is like kind of a big group of folks. So, lots of people from, not just Georgetown, but NASA Goddard and UT and Carnegie and Glasgow and Harvard and Penn State and Santa Fe Institute and Toronto and Carnegie. And I should say, we're certainly not the first people to sort of be thinking about these issues. I feel like we're really building a lot on seminal work that's been done in our community. Even the very first issue of Astrobiology had this great article by Pan Conrad, as well as Ken Nielsen, talking about, how can we look for life as we don't know it? And we've got John Barros and Joy Holer and, all these other folks that I've listed here that have also been doing some really exciting thinking about this topic, this idea. But so I'm going to tell you about some of the kind of specific ways we've been kind of pushing on this question. Again, the sort of central question is like, if we really want to be mission relevant, if we want to think about the sorts of things that we could turn into instruments one day, what are the tools and techniques that we could possibly develop that really do fit this criteria of agnostic? And so there are 4 areas. I mean, there are many more than this, but there are 4 areas that we've really been focusing on. And I will walk you through each of those. So one is kind of molecular pathway complexity, molecular complementarity, chemical fractionation, and disequilibrium energy transfer. So let's start off with this very first one, this kind of idea of pathway complexity. And is this working? Oh no, is this video? It's probably jogging for you guys too. Let me try that again. See if I can get it so it doesn't jog. Okay, that seems a little bit better. Can you sort of see that there? This is just this little animation. And so you can sort of think about like some beautifully, carefully sculptured statue and like the sort of molecular kind of equivalent to that. So as my colleague Lee Cronin over at University of Glasgow, he likes to say, you know, if you found a 747 on the surface of Mars, or maybe the sort of more apt analogy now would be like a Tesla Red Roadster. I don't know, you find something really complicated on the surface of Mars, and especially if you found like a whole fleet of 747s, or the whole set of really complicated, complex molecules, you might have just no idea how they possibly got there, but it probably wasn't just random, right? And so if you try to think about this like a little bit more concretely, like in a molecular context, you know, there's this idea that you could, you know, maybe if you've got random processes, you could sort of generate like a sort of one-off instance, you know, something. But if you find a lot of these things, as I've seen before, it could just be really, really indicative and interesting. But most of the ways that we have thought about complexity, you know, as a community, have been like a little bit subjective. Like I say, well, this molecule seems, you know, complex, but they're just, you really need like a metric, you know, some sort of robust criteria. So the question here is really, can we define this threshold of chemical complexity that is unlikely or maybe even impossible to form spontaneously? And it's really based on this thesis that, like, if you've got a living system that's, say, replicating or evolving, like, you're going to generate all of these complex molecules. You'll get things like metabolites and cofactors that would just be really highly unlikely to form in any significant quantity in the absence of biology. And so I said, like, it's been kind of subjective how people have defined complexity for a lot of this time. And so most people, I think we could all agree, if we have some, you know, little molecule like methane, like shown here, like, yeah, we can probably all agree that methane is like a pretty simple molecule. And then you can think, well, what could we do to this molecule to add some complexity? So maybe some different types of bonds, you know, like if you get different types of bonds, then things will be more complex, and as with, like, say, a higher molecular weight, you know. But there's this thing that happens too, where you get polymerized molecules, and they can have different types of bonds, and they can have a high molecular weight, but maybe they're not so complex because they're just repeating subunits of the same thing. But then, like, once we start seeing all these different components together, different bonds, different types of elements, structural features like branching, like rings, like heteroatoms within those rings, like we're starting to feel more complex. And so I think this is the sort of gut intuitive sense we have about this whole thing. But we can also, like, be even more concrete about it by using algorithms. And so Lee and Stewart Marshall and others, you know, publish this cool paper, you know, that is talking about how you can use graph theory to answer this question. And so there's like this long history of using graph theory for sorts of things, but the key question here is, what's the simplest way to construct a molecule from all of its parts after you've accounted for that simplifying feature of duplication? And so, like, you could use this sort of graph theory algorithm. So here's just a simple example. You've got biphenyl. It can be broken down into 6 steps. And you'll note that if you're just repeating a unit, that doesn't count as a new step. And you can assign a complexity index. And in this case, it's 6. Because it takes 6 different, like, very distinct different things have to happen to, like, create this molecule. And so you can do this for all kinds of molecules, and you'll see these, like, simple things like glycine are easy to make, you get a low index, but things like morphine, ATP, like, these start to get more complicated. And what's really fascinating is that it seems to be that, you know, without making any assumptions about these kind of underlying chemical structures, there seems to be this threshold of around 15, like a molecular assembly index or pathway complexity index of 15, above which these molecules are really unlikely to form without supporting biological machinery. And so here is this Nature Communications paper that just came out last year, really starting to explore chemical space. And you can see this like really holds up in this really interesting way. So of course you can always miss indicators of life below this threshold, like DNA, like our information polymer, like storing all of our genetic information, like that is a repeating polymer. And like, of course, that's a great biomolecule, a great biosignature, but like, there are other things like that are simple that we may miss. But there are all these things that are complicated, that are biological, that are above this threshold. And so this cool paper, again, is sort of showing that, once you go out and you start exploring chemical space, even with blinded samples that they had no idea what they were, you see that things that are kind of inorganic or abiotic are all clumping down here. And that once you get past that, you get this whole world of biological molecules, things like ATP that are, when you sort of go through this whole thing, increasing the molecular assembly, you get up to these thresholds that just seem really exciting. So basically saying, if we found something like ATP, if we found it in significant quantities on the surface of Mars, like it would be very, very powerful as a biosignature, or at least a biohint, shall we say. But of course, one of the key challenges is detection, right? You have all these different things that you get to see, but where the rubber hits the road is like, can we really do it with the kind of flight capable suites of instrumentation that we have available to us for planetary missions? So another big part of our project is trying to translate the kind of fragmentation mapping using MSMS and testing it on flight-capable ion traps. So, this is one of the reasons I'm so disappointed about the sort of delay of the Rosalind Franklin rover. It was supposed to be European rover launching on a Russian rocket. And so first that's been delayed because of the conflict in Ukraine, but I really, really want to see this wonderful mission go. So MoMA is one of the instruments, and this is kind of like a bigger, well, it's actually not bigger, it's smaller, but a better version of the sample analysis at Mars, the SAM instrument that we've gotten so much great data out of with Curiosity. So MoMA is like not very big at all, in fact, but it incorporates a gas chromatograph mass spectrometer, GCMS, as well as a laser desorption mass spectrometer, an LDMS, into this like very, very, very capable instrument. And it has this ability to sort of, I'll tell you in a second, just more about the MSMS capability, but it's allowing us, it's really the first time I think in situ we'll be able to sort of, incorporate some of these things like molecular assembly and test them on another planet. So just a really quick word about how amazing, I just think this instrument is so great. You get this like massive organic range. You've got this laser source, this 266 nanometer laser. And so you get like your laser can generate these plumes of ions. They're transferred by the gas flow into this miniature little linear ion trap mass spectrometer. And it's like similar to a thermo LTQ, like a multi-LTQ, which we have in the lab, which we just retrofitted with a 266 nanometer laser. So we'll be able to do really cool high fidelity benchtop analyses that will sort of hopefully help interpret this data when this mission does eventually go. But you see, you can use pyrolysis, sterilization, as well as this laser desorption mode. And you get this huge sort of range in terms of molecular weight. So MoMA can also, I think this is great. So perchlorates on the surface of Mars are just so tricky when you heat them up. They can be just kind of erasing your organic signal. Perchlorates have been really complicated when we've been trying to do GCMS on Mars. But because of this laser mode, where you're not actually using heating, you can sort of still detect these really heavy organic compounds, even in the presence of perchlorate, which is so exciting. And then you've also got this MSMS mode. So you can take any sort of single peak and you can you can target that and select it out and refragment it out. And so I'll show you here. Let me just skip ahead really quickly. So this is just an example. So this is using ion traps that are similar to MoMA. And this is the MSMS data from just one particular peak. So you can take this 825 Dalton paraformaldehyde ion and you can refragment it, and you can see clearly these sort of repeating, mostly identical mass losses. And so that indicates like, yeah, this is a polymer. It's got high molecular weight, but it's got low complexity. And so there's all this work that we're doing, really trying to build a training set of data with prototypes and breadboards of these flight capable instruments. And it's not just MoMA, it's also DRAMs. And so Dragonfly, our exciting quadcopter that's going off to explore the surface of Titan. It also has a mass spectrometer, this instrument called DRAMs here. And it's housed up here inside the quadcopter, but it's got a lot of similarities to MoMA. It's also built by the same team at Goddard, working in partnership with the French, and it's going to be really another really exciting instrument, another really exciting opportunity to kind of test some of these theories about molecular assembly. Okay, so we're doing lots of work there, and I'm gonna move on now to like another example, another one of our theme groups here as part of this ICAR project. It's kind of in some ways along similar lines. It's looking for complexity of surfaces, kind of in this sort of roundabout way. So let me sort of back up a little bit, and I know like lots of folks like Jordan and Jose Luis and Chris Carr and other folks have been really excited about looking at what nanopore sequencers can do in terms of sequencing for spaceflight. So I really think it's like one of the most exciting things in the world of genomics these last few years is that there are these incredibly small handheld sequencers that have been developed. Again, rapid advances in miniaturization have kind of made these kinds of things possible. So here is this commercially available instrument that's just like all of the work. Even we spend years taking these huge benchtop instruments and trying to get them down to these little tiny things that we could send to space. And they've already just done so much of this work, sort of developing these platforms that, this is 10 centimeters long, it weighs 85 grams, it drives only a watt of power. And it's really exciting. I think it's incredibly exciting for like field research and understanding things like the limits of life, being able to get immediate data from places that you're working. I think it's also going to be really great for minimizing environmental impact. A lot of us that work in extreme environments, worry about sort of taking too many samples and the impacts that you leave behind. But if you can do a lot of triaging, you don't just have to collect everything under the sun. You can kind of have a sense of what you're doing. I also think it's great. I mean, this fortunately has not happened to me, but like sample loss, like if your samples thaw or they get stuck or they don't make it back, you could like lose an entire season of field work. But, if you could take your, hard drive with a bunch of data already on it and throw it on the plane with you, you could at least have kind of like a minimal amount of sequencing data already, which is exciting. But anyway, I'm turning into like an advertisement for the company now, so I'll keep moving. But it's this teeny tiny thing. It sequences native DNA, so it avoids PCR bias. It's pretty hardy. You can use it far from electricity under arduous field conditions. Though sometimes its performance is enhanced if you add little hand warmers. So just kind of backing up for those of you that don't know, the way this works is the instrument is measuring ionic current as the DNA passes through a protein nanopore that's embedded in the special polymer membrane. And as the DNA strand is sort of coming through, it causes this characteristic current change for each group of nucleotides. And that's kind of how you do the sequencing. But you'll notice I say it's a protein nanopore. So this is actually a biological protein nanopore. So, we've had really exciting NASA-funded efforts to develop sequencing for life detection to look for things like RNA and DNA. And it's like, well, why not? Why would we not do that? There's so much material that was potentially exchanged. Well, we know it was exchanged between Mars and Earth early in our histories. We could have shared ancestry. We know like nucleobases that found meteorites, they could be widespread in the solar system. And like you just get so much information, you can pen that onto a phylogenetic tree, you can figure out like where it diverged, where the last common ancestor was. And so lots of really cool efforts sort of being developed under NASA's Matisse and Cold Tech instrument programs, using both kind of the biological nanopores, but also solid state nanopores, which could be more robust, eventually, but still needs some more development. And so, yeah, you've got your little pores and they're arrayed in this like microscaffolds that are implanted into the sensor chip in the integrated circuit. One of the things that's been pushing for sort of more of a solid state nanopore model is this idea about sort of radiation, stringent temperature limits and shelf life. And you get these things from the company and they say, oh, you can't use these you got to keep them at this particular temperature. You got to keep them for, like they'll expire after a year and, all these things that are, that make it seem like they're not going to be very stable. But, we've already tested these in space. And then Mark, who's this guy who was working in my lab for a while, also wrote this really cool paper about the radiation tolerance of nanopore sequencing technology. And like even the protein nanopores, like even these things that you don't think would actually be so robust seem like totally fine for a mission to Mars. You'd have to really think about it for Europa and what kind of radiation shielding that you could do. But both in terms of longevity and also the sort of radiation doses, we used to source at Goddard and expose the hell out of these things. And so it just seems very clear that this is a promising potential future technology. You could monitor contamination, which of course is a key challenge with any kind of life detection. You could look for nucleic acids, like including those with non-standard bases like XNA and potentially other charged polymers. And so we're starting to get agnostic even just with this setup. And then one thing I wanted to talk to you about too is this idea of molecular complementarity, which is one of the things that has gotten me sort of most excited about sequencing for space. And this is this idea of using these little small single-stranded short oligos, like sequences of DNA or RNA that are short, 15, 30, up to 70 base pairs in length, and not even necessarily that full length, but like some part of that combined, some portion of that combined. And so if you can harness the power to all kinds of ligands, I'll show you here in a second. And so if you can harness the power of sequencing to explore sample complexity as well, so not looking for reverse complement binding, but looking for just all kinds of different binding patterns, all kinds of different things, and take kind of a big data approach to it, we might be able to do, and it seems like we will be able to do like some of this really cool, just sort of looking after this idea of surface complexity. So let's back up just a little bit. So we're kind of borrowing this idea that's been around for like a while in the world of cancer biology. It's called systematic evolution of ligands by exponential enrichment. And so like, let's sort of think about this kind of this idea that you could take this random pool of oligos, those little short single-stranded oligos, and so you've got like millions and millions of them, and they're just randomly generated, this big library. And so if you take those and you incubate those with a target cell, say you're trying to find breast cancer cells in the human body, and you incubate those like, in the lab with some target cells, most of those are not going to bind. Some very, very, very tiny portion, way less than 1%, is going to find some place on that cell that it binds. And so you can go through this process where you collect these sequences and you kind of go through to kind of get rid of this non-specific binding, and then you can amplify those. And the idea is like at the end of all of this, you can find a couple of things, a couple of these little short oligos that can be kind of great reporters. And you don't necessarily know what they're binding to on the surface of your target cell, but they're binding really specifically. They can bind with similar specificities and affinities as antibodies, but again, they're made of nucleic acids as opposed to protein. And so we've long thought about, like the RNA world and this possibility of like RNA binding to clays like montmorillonite. Those were probably really important as templating surfaces early on around the origin of life. And it's not just minerals like clays that these little single-stranded oligos will bind to. Like they'll bind to all kinds of things: peptides, proteins, small organics, minerals, metals. Here are a bunch of these articles sort of going through all of these different binding properties. So if you take like a really, really large library of these, because these nucleic acids sort of naturally curl up and they have secondary and tertiary structure, like you get some binding. Even if it's sort of weak binding, you get this binding. And so this is kind of the idea of this molecular complementarity approach. So this again is kind of some toy cases. You imagine like just a cell, even if it's a really primitive cell, you can just imagine like there's this vast amount of information that's patterned on the surface of even those primitive cells here on Earth. And, on the other hand, like say you've got like a mineral grain, like the sort of theory behind this is that, if you've got sort of a repeating crystalline structure, sure, there will be binding sites, but probably not as many, and is not as many different binding sites as, say, a cell surface. Especially over very, very small, you could do serial dilutions, you could get down to like sort of single cell, single particle, and over those like small surfaces, like that there would be more and more varied binding sites on the surface of a biological cell. And so this is kind of flipping this process of CellX, because we're not pre-picking the targets. We're not saying, here's a breast cancer cell, go find it. We're saying we want to analyze this full pattern of binding that results from using a sufficiently large, randomly generated library, and then look at the sort of results. So again, these are not binding to like other nucleic acids. Like you don't need to have DNA or RNA-based or nucleotide-based life, but if you had some sort of a very, very complex structure that was created by life, especially with lots of complexity over small areal extents. This is kind of our theory going into this. So, initial work kind of in this world of aptamers has shown, working with these little oligos that with no prior bias, you can really very reliably pattern differences with this big pool of these randomly generated oligos over samples that have really, really similar cell surface attributes. So these are just different cell lines. But of course, for us, the target is like complexities, anomalous patterns, like where we see lots and lots of complexity. So yeah, to sort of explore the statistical space, we've got lots of different statistics that are sort of coming into this. And one thing we've been doing in the lab, so this work is primarily being carried out by some of my students at Georgetown, as well as at University of Texas with Andy Ellington and Eric Anslin and some postdocs and students down there. And one of the really cool things is this thing called proximity ligation assay. So we've been doing some experimenting with this. So basically, we're taking two different probe libraries, and they've got these random regions of random, like, so the nucleotides are random in the middle, like these little short oligos, random in the middle, but you've also got these probes sort of on the end of them. So these ligated probes, so they come together with a splint, as you can see sort of right here. And you get the forward primer on some of these little, on half of the library, and then you get the reverse primer on the other side, and you've got the splint that will join these things together. And so this requires that two different probes come close together in proximity to one another on a cell surface, but only those that come together and can be joined by the splint can actually be amplified. Because when we use PCR, the polymerase chain reaction, to amplify up this signal from a tiny, tiny little signal to like a big, robust signal that's easy to read. And the reason that we've been trying to do it this way is because it's just exquisitely sensitive. So you basically have to have these two different DNA libraries, and they're interacting with a material, and because we're thinking about a lot of this in the context of sort of ocean worlds, liquid samples, the oligos that are not bound to the substrate can be sort of washed away, but you get exquisitely low background. So, it's really rare for like two of these things to come together with a splint if they're not actually in proximity to one another because of a surface. And so that has been a really fun approach. And so what we've been doing is going through this ligation process, wash process. We go through the PCR amplification. We sequence that product after we excise it, and then do data analysis. So we can do these washes to sort of remove these unbound probes, and then we have selected amplification of these ligated sequences only. And so coming soon, a paper on all of this and our first data results, looking at lots of positive controls and substrates. We hope to have that out shortly. And one of the things I'm so excited about is that this is it's so good for low biomass targets. Chemolithic autotrophic or chemolithic trophic life on ocean worlds. Some of the models that we think about, like, Tori Holler, for instance, has done a lot of work suggesting that, we might have really low biomass, the energetics might be such that, we're not talking about a lot of life available for us to detect. But because these oligos are made with nucleic acid, we can potentially do PCR on them again and get like a big signal to noise, which I think is really great if you've got low biomass worlds. And so this, I think, would be great. You send some sort of a small sequencer to space, you can look for life that's, DNA, RNA, potentially XNA, maybe even charged polymers. You can look for contamination, then you kind of get this trifecta too, because even if it's none of that, you can still maybe use this to look for evidence of sample complexity, which would be a really cool agnostic biosignature. But even if you can't send your sequencer, like even if for some reason that's too complex or the data is too much, there are these alternative detection possibilities. You can, like, these simpler setups could also work. You can have microarrays with spots that contain multiple strands of a single DNA sequence, and you could have, millions of those arrayed on a glass slide. Something smaller than a glass slide, and fluorescent probes, which could be released with binding. It could be simply imaged. You maybe even do electrochemical microarrays with electrochemical reporters like ferrocene blue. Really excited. So I want to leave lots of time for questions. So let me speed up a little bit. There are still a couple more things that I wanted to talk you through. Another big theme of our project is this idea of elemental and isotopic accumulations. Like if you could have accumulations in compartments that are isolated from the environment as potential biosignatures. We've got all these new tools, like we've got nanosims for biosignatures and sims for isotopes, and LAICPMS, to look for elements, and TOSIMs for more bioimaging. And you can use all of these really cool sort of new tools to really look and see, is there some sort of compartmentalization that we can see. And so we're doing lots of work. So this part of our project is being led up by Chris House at Penn State and his amazing lab group, thinking a lot about using complex fluids to grow up microbial cells, and then in parallel, these abiotic, and really trying to trick ourselves with how many sort of abiotic concentrations can we get in terms of these compartments. But if we have these kind of isolated structures, it can be really exciting, again, taking advantages of lots of advances in technology. We also are working with Jeff Marlowe at Boston University, thinking about patterns of fluorescence, and are there spatial arrangements of fluorescence that are more likely to be autofluorescence versus cells, mining this huge atlas that he's built of kind of metabolic activity and community mapping as part of this big project. And so many imaging of cells on different substrates like deep sea carbonates and volcanic fumarole sediments, and modeling what kind of arrangements emerge. We've also been, this works at different levels, right? So at the micro scale, we sort of at least, have this sense that life can concentrate scarce elements. In our case, here are some examples into these discrete entities that are geochemically distinct. And so we see that, sort of looking at, cells are enriched substantially beyond, say, just abiotic precipitation of calcite from seawater. And so that works at the micro scale, but we also might be able to potentially detect stuff like this at the macro scale, just kind of this idea of looking at, we have this really specific red field ratio for the Earth, the ratio of carbon to nitrogen to phosphorus and marine biomass and all the dissolved nutrient pools. We have this very, very nearly constant 106 to 16 to 1. And we can see that biology results in this biosignature, this global, pretty constant biosignature, just even at the bulk scale of seawater, even after the death and decay of the actual microbes. And so we've been thinking about elemental abundances also, like, could there be rare field ratios of exotic fluids on other worlds? How could we model those and think about those? But wouldn't that be neat, if that exists? And how could we think about that as another kind of cool potential biosignature? So as I was mentioning, ongoing work, like really looking at like the elemental and isotopic composition of cells versus other, like these authigenic minerals and other precipitates. Like this is down in Kid Creek, this really deep mine in Canada with subsurface pore waters. Lots of SIMS work and other LAICP-MS work being done by the house group on this. Okay, and then finally is energy transfer. Just love this quote that I threw up there: life is nothing but an electron looking for a place to rest. So we know that microbes can utilize solid phase minerals as electron acceptors, like insoluble ferric oxides. And so you can utilize like an active anode as an electron acceptor the same way that you utilize something like a insoluble ferric oxide, as an electron acceptor during aerobic respiration, during respiration. And because the response, it actually is very sort of distinct. And I'll show you here in just a second. So we're seeking disequilibrium redox chemistries that are inconsistent with abiotic redox reactions as an indicator of active metabolism. This work is being really pioneered, and our point person for this theme is Pete Gerges up at Harvard University. And really trying to look at the electrochemical attributes associated with abiotic oxidation versus biology. And so this is really evident even in like the simplest of setups. So this is just this really cool experiment that was done by this group over in China. These are looking at huge tailing dams. So like you've got these iron sulfide tailings as a byproduct of mining activity. And this group was like, oh, maybe you could turn this into like this huge microbial fuel cell. And so they set up some sterile controls. And you can see in these sterile controls, which are the sort of darker dots here, that you get abiotic oxidation, but then the signal, it really rapidly falls away. But in these inoculated chambers, especially after a while, you start to see that each time the iron tailings are added in, the biology responds differently. And you see this like really characteristic sustained signal, this marked and sustained increase in voltage in the reactors where you've got microbes there. And so like this is, like when we think about mass specs, we've got super high TRL, we send those out to space all the time. We're just kind of thinking more about how can we look at the data in new ways, but this is like a much lower sort of TRL or technology readiness idea. But like, could we someday like come and like stick something at the sediment water interface in like an ocean world or something, and actually look for, electrochemical signals of life, again, without sort of presupposing anything about the molecular components there, the biochemistry. So anyway, Pete's group is doing some really cool mesocosms using mostly iron oxidizing bacteria with like varying conditions, and sort of, laying that out, like with the abiotic setups where you get the autocatalytic oxidation, but in the biological one, you get both, but you do see again, this kind of sustained and marked increase. And so the goal again is developing some sort of, in situ probe that could potentially work in this way. Okay, I will finish up quickly. Just to kind of add on, like, we're working with some really fantastic people at the Santa Fe Institute, like Chris Gimpes and Eric Libby, thinking about data analysis. Because one of the things is, like, if you have like these methods are inclusive, but this inclusivity, there's a bit of a trade there, because it's not going to be, if you find chemical complexity or electrochemical signals, as definitive as like a DNA sequence or a hopane, like we are being more inclusive, but, we might not be as definitive as some of these things. And so we've really been thinking about how can we approach this in a way where we could combine multiple approaches. And fortunately, when we send spacecraft off into the off into the solar system, we're able to usually send not just one instrument, but a whole suite of instruments. And how can we design astrobiological suites of instruments? And how can we really use all of this data that we're getting back in and combine it all? And so lots of interesting work with Bayesian networks where you can really think about outputs in terms of probability. There's a biosignature, given that data, really thinking about measurements being converted into likelihoods and thresholds. So some really cool work doing that. And then finally, really just trying to move. I mentioned this when we were talking with the grad students, this idea, I think we have this idea, it'll be this sort of eureka moment. We'll be like, okay, that's life, and we'll be for sure, or no, that's definitely not life. And I think what's probably more likely, especially when we're in very foreign types of planetary environments, is that it would be better if we could say something like, okay, this is 3 sigma away from what we would expect from abiotic processes alone, and sort of thinking more along a conceptual spectrum of certainty. And all of this work is really focused on what can we do in situ, what can we do sort of here in the solar system. I'm partly kind of a restless sort of person, and so I think that that's an exciting way to think about it. But of course, we have over 5,000, I think it's 5,100 now confirmed exoplanets orbiting distant stars. And of course, even though this project has been mostly in situ focused, there are all these some of these techniques can really apply to remote sensing measures as well. And so we've been working a lot with the Enfold, Network for Life Detections also does a lot of cool work with lots of overlaps with the Nexus for Exoplanet System Science, or Nexus is an acronym within an acronym. That's another one of the RCNs. But anyway, happy to talk more about that if people have questions. And then the goal, of course, is to come up with something like a tricorder from Star Trek to really be able to go into this environment with a really robust suite of instruments. It would be great if we could look kind of specifically as well as more broadly. And I will say that there are lots of ways to join us if there are any undergrads or grad students thinking about opportunities. We've got this website. It's hard to read there, but it's www.agnosticbiosignatures.org. And we've got lots of summer internships, and we post postdocs and graduate opportunities there too. And I just want to say finally, thank you to my lab, including sweet Chad Lazarski, who's here in Georgia Tech, who also got to work on lab and was a great contributor. And yeah, none of this would be possible without all these extraordinary people that are doing all the work. And I'm stopping, and I'm sorry I went so long, Tatiana. But hopefully we still have 20 minutes for questions. And if people have questions we don't get to, you can always email me.